Okay. Last time we stopped here, right? So what do you find in plant cells? Plant cells you find all plant cells have. Let's say it's a plant cell. Okay. This is a large vacuole. What's the name of the membrane? Tonoplast. Yes. Mm. This is called tonoplast. What's the liquid inside? Cell sap, right? What is cell sap made up of? Water, glucose, amino acids, and waste products. Okay, it's like storage. But some animals do have vacuoles. For example, do do you remember amoeba? You studied in class 9, right? Amoeba. Where do amoeba live? It lives in water. So water keeps entering the amoeba. What will happen to amoeba if water keeps entering? It will burst, swell and burst. Why is it bursting? There's no cell wall to protect it, okay? It can burst. So what do they have? They have a special kind of vacuum known as contractile vacuum. They are like our kidneys. Suppose you drink like one full bottle water, within 10 minutes we produce urine. Because you don't need so much water, right? It's not good for the body. So the excess water is collected by contractile vacuole and release it outside. Okay. So what is this? Contractile vacuole. Where do you find? You find in animals that live in water, like amoeba, one-celled animals. Okay, not in the large one single-celled organism. Do you know this organism from class 9? Yes, Salome.
Yes, sir.
What's the name of this? This one.
Um,
Do you know this animal?
eh,
Yeah, it's
no,
called
sir.
paramecium.
Okay.
Paramecium, right? Okay. And even this has contractile vacuum. Even this, here you find contractile vacuum. They don't have cell wall. Okay. Plant cells don't burst. Why plant cells don't burst? They have cell wall. You know, algae. Do you know algae, right? Algae is a type of plant. Some of them have cell wall, which prevents bursting, right? Why do animal cells need this vacuum? To prevent bursting. Okay, to prevent bursting, it needs a vacuum. Is it clear? So, what's the function of contractile vacuum? Excretion of water. And why it is excreting water? To prevent bursting. And why is it bursting? Because they don't have cell wall. Okay, they're animal cells, they don't have cell wall, so they must get rid of water. Can you tell me why water keeps coming in? Okay, because it's by osmosis. Do you remember osmosis? Yes. In osmosis, water moves from high water concentration to low water concentration. Okay, and that's why water will enter. And but to prevent bursting, what do? They pump the water out. What's the name of the vacuum? Contractile vacuum. So where do you find contractile vacuum? Animal cells. Where do they live? Water. What's the problem for these animal cells when they live in water? And water keeps entering by osmosis, right? What will happen? Why? Cell wall, right? How do they prevent bursting? Contractile vacuole. Contractile vacuole. What do? Contract water. It removes excess water and prevents bursting. Okay. This is the next cell organelle. Okay. How many membranes around vacuole? One. Lysosome, Golgi, endoplasmic reticulum, ribosome, none. Okay. Right. Now this has two. From now onwards, you will find two. Right? This is called mitochondrion. Always we say mitochondria, right? This is singular. You talk about one mitochondrion, not mitochondria. Means many mitochondria, right? Okay. This one has two membranes. Can you see? There are two membranes here. These are the two membranes. What do you call these membranes? These are outer membrane and inner membrane. This is inner membrane, that's outer membrane. So how many membranes around mitochondria. There are two membranes, outer membrane and inner membrane. So, what's the difference you find here? The outer membrane is smooth, okay? It's smooth and continuous. But look at inner membrane. It's folded into finger-like structures. This finger-like structure is called cristae. What is it? Cristae. Okay. Why is it folded inside? Like why there is mesosome in bacteria? To increase surface area. Similarly, if this increases surface area, do you understand what does that mean? Increase surface area, right? How about you? Means what? No absorption, but the idea of increasing surface area, does it make sense? Like, suppose, and I'll give you an example. Suppose you take a thread and measure this line, okay? Length of the line. Then take another thread and measure the length of this line. Which one will be longer? Inner one, isn't it? So the membrane is folded up, means it has more area inside. Okay, that's the idea. So inner membrane is folded into what? Cristae. Okay. Between outer and inner membrane, you find a space. This space is known as intermembrane space. What is it? Intermembrane space. This space is for storing temporary storage of H plus. What is H plus? Protons. Okay. H plus is protons. So it's a function of intermembrane space to store protons temporarily. This is in more detail in another chapter known as respiration. So I won't explain everything because we have to do there. Okay. There are more things. So how many membranes around? Two membranes. What are two membranes? Outer and inner membrane. What's the space between the two? Intermembrane space. Okay. And what's the function of intermembrane space? Yeah. Storage of what? Protons, H plus ions. Okay. And what's the difference between outer and inner membrane? What's the difference between outer membrane and inner membrane? It's folded. What's the name of the folding? Cristae. Okay. On the cristae, you find some structures. Okay, there are different names for this. One is called elementary particles. Okay, that's one name. Second name is stalked particle. Okay, what's the second name? Stalk particle, right? Or stalk particle. And what's the function of this? This helps in the formation of ADP. Okay, ATP. You know ATP, right? Do you know ADP? Do you know ADP? No, diphosphate. Okay. Do you know ATP and ADP? Yeah, ATP is a source of energy. It's produced from ADP. Okay. This is adenosine diphosphate, right? If I draw this is adenosine. When it has two phosphate, one, two, three phosphate, what do you call it? This is ATP, triphosphate. And when it release one phosphate, it has two of them. And now this is called ADP. Okay. And when ATP changes to ADP, energy is released. For example, when you do exercise, what happens to the muscles? Contract. It needs energy. Where does energy come from? ATP. But in the process, ATP changes to ADP. Okay. And when you rest, respiration changes ADP back to ATP. Where? In the mitochondria. Okay, in the mitochondria, respiration happens, and on the elementary particle or stalk particle, the ADP is converted to ATP. Okay, what's a jelly-like substance inside? This is called matrix. What is it? Matrix. Okay, Matrix is a movie, right? Game movie, right? Anyway, matrix. What's a jelly-like substance? Matrix. Okay, in the matrix, you can find there is a circular DNA. Okay, there's a circular DNA. Where do you find circular DNA? Bacteria. Okay? How come it's in the mitochondrion? Mitochondrion was a bacteria almost 3 billion years before when the life originated. This bacteria got into eukaryotic cell and it started, getting food from the cell, and it started giving ATP to the cell. So it stayed. Even today, we can see the evidence. What's the evidence? Presence of circular DNA. Okay. Plus 70S ribosome. Where do you find 70S ribosome? In bacteria. See, even you can see it has its own DNA. No organelle has its own DNA except nucleus. That's our DNA, isn't it? But this is the DNA of mitochondria, and cell cannot form a mitochondria. We actually get all mitochondria from our mom's side. Okay, the egg cell has cytoplasm. It has like few mitochondria. That's what we inherit. We don't produce new mitochondria, and it divides like a bacteria. So, what's the evidence that it's a bacteria? It has circular DNA and it has 70th ribosome, and it does its own protein synthesis. Okay, it doesn't have to depend on the other cell. And what else you find here? Okay, this one has enzymes. Enzymes for aerobic respiration. There's a name for this. I'm not saying that now because it's in another chapter. It's called Krebs cycle. Enzymes for aerobic respiration. Okay, it has enzymes for what respiration? Aerobic respiration. Make sense? Okay, there are more details, but we won't study anything now. We'll study only this. Okay, so what's the name of the cell organelle? You'll keep using this term. What is organelle? Organelle means what? Organs of the cell. Parts of the cell are known as organelle. And what's the name of this organelle? Mitochondrion. Why mitochondrion? It's only one mitochondrion. How many membranes around? Two. What's the difference? Name the membranes. Outer and inner membrane. What's the difference between the two membranes? Is folded into what? Cristae. What do you find on the cristae? Elementary particles. Okay. Do you know if you do exercise and go to gym or play games, your mitochondria gets longer. Do you know if you keep playing, you get more stamina? Okay. Means more energy. If you don't do anything, your mitochondria get smaller, and you also develop more cristae. That means you produce more energy. You remain active all the time. That helps in your studies also. Okay, so if you get time, you can do some exercise. Okay, that's anyway. What do you find? Why is it finger shaped? Why crystal is finger shaped? Why is it finger shaped? Increase surface area. What do you find on the crystal? Elementary particle. What's the function? What's the other name? Stalk particle. What's the function of elementary particle? Yeah, formation of ATP from ADP. And uh, and what do you find in the matrix? Matrix has enzymes for aerobic respiration. Matrix has circular DNA. Like bacteria, matrix has 70th ribosome, like bacteria. So what are two evidences to prove that mitochondrion originated from bacteria? Presence of 70th ribosome and circular DNA. Is it good? Okay. How many membranes are on mitochondria? Two. Name the membrane. What's the space between the two? And say the difference between outer and inner membrane. Hmm. Called cristae. Hmm. Increased surface area. What do you find on the cristae? Particle or elementary particle. And what is its function? Hmm. ADP. Okay. Salome.
Yes, sir.
How many membranes around mitochondria?
Two membrane,
Can you name them?
inner membrane and outer membrane.
What's the difference between the two? What's the difference between the outer and inner membrane? Inner membrane is folded to what? Folded into cristae. Okay? Yeah.
Christian, yeah.
Yeah. Samaira, what's
Yes.
similarity between mitochondria and bacteria?
Uh, they have a circular DNA and
And
ribosome.
70s ribosome. Okay, 70s ribosome. And these are the sizes. Okay, length and size. This you have to learn, especially for NEET exam. You must memorize it. Okay, I'll send you the notes of that also. Okay, so that's they're talking about the structure, matrix. Okay, all those 70s function. What's the function? Yeah, aerobic respiration. Never say this. This you should never say. In small classes, they used to say powerhouse of the cell. Don't say that. Okay, you must say the specific function. What do they ask? Produces energy ATP by aerobic respiration. Okay. The next part is plastid. Heard about plastid? Yeah, what is plastid? Something related to what? Chloroplast. Okay, you know chloroplast, right? But this is like a common name. Like I say, students. A student can be a boy or girl, isn't it? Similarly, when I say plastid, there are two kinds of plastid. Some plastid are colored. Some plastids are colorless. Okay? Colorless plastid. Again, we have a lesson on photosynthesis. Full lesson on photosynthesis only. There we'll study more detail, but only basics in this chapter. Okay. Colorless plastid does storage of food. Okay. Colored plastid perform photosynthesis. So plastids are of two kinds. What are two types of plastids? Colored plastids and colorless plastid. Okay. They call the colored plastid chrome means color. Google Chrome, color, right? Chromoplast. Chromoplast means colored plastids are known as chromoplast. And colorless plastids are known as, okay, the colorless plastids perform storage of food. Like it can be starch, it can be protein, or it can be fat. If it's starch, they call this amyloplast. Okay, amyloplast. Protein is called proteinoplast. If it's fat, this is called elioplast. Okay, and what's the name of this plastid? This is known as leuco. Do you remember leukocytes? No, white blood cells. Okay. Leuco means white. Leucoplast. So what is leuco means white. What is chromo means colored. Okay. Chromo is colored and leuco is white. Okay. It cannot do photosynthesis, but this can do. One example for this, if chromoplast has green color, this is called chloroplast. Okay, but there is another name for red plastid. Have you seen leaves which are red in color, yellow in color? Each one has a different plastid. That's the reason. But we study only this one here. The other, rest in others chapter. If it's green colored, it's called chloroplast. Why is it green? Because it has chlorophyll. Okay, it contains chlorophyll and it does photosynthesis. Make sense? What are two kinds of plastid? Colored and colorless. What's the name of colored plastid? Chromoplast. Chrome means what? Color. This other one, leucoplast. What is it? Colorless. What's the function of colorless plastid? Storage of food in the leaves, in the stem. And what's the function of colored plastid? Photosynthesis. Where do you find this the most in a plant? Where do you find this most? Which part is the most green in a plant? Hmm. No, surface of what? Yeah, leaf, right? If you remember, leaf has cells known as palisade cells, mesophyll cells. Okay, and this is chromoplast. You find most of them in leaves, right? This you find in storage organs like potato. What do you find in potato? What's the Stored food in potato, starch. Starch. So it has more amyloplast. Can you name a nut which has a lot of protein? Protein rich nut? Mhm. Yes, walnut or cashew. Okay, more protein, it's even tastier, right? Okay, and in these nuts you find proteinoplast. And name something, a seed which has a lot of oil. Hmm. Sunflower. Huh? Why you're forgetting your coconut? Yes, coconut is our nut, right? So, like for example, coconut, there's plenty of oil, right? Mhm. It has elioplast. Make sense? Okay. So these are the types of plastin. What are two types of plastin? Colored and colorless. What's the name of colored plastin and colorless plastin? Why is it colored? What? Chlorophyll. It contains chlorophyll. Remember, okay, chlorophyll. And what's the function of chloroplast? Where do you find most of them? Leaves, leaves for photosynthesis. Okay, and can you name the colorless plastin? And what do they do? Why is it white in color? Because there's no chlorophyll. No chlorophyll. Okay, and if it stores starch, what do you call it? Amyloplast. Do you know amylase enzyme? Amylase digests starch. So it's if it's related to starch, it's amylase. Second is easy, protein, proteinoplast. But elio means fat, lipids. Okay. So what are three types of leucoplast? Amyloplast, proteinoplast, elioplast. Okay. Which one store fat? Elioplast. Okay. Which one store starch? Amyloplast. Other one is easy. Okay. Yeah. Samira. Name the two types of plaster.
Colored plastic and colorless.
Okay. Name the colorless plasters.
Colorless is leukocyte.
Okay. And where is she? Salome.
Yes, sir.
And name the colored plaster.
Chromoplast.
Chromoplast. And what? Why is it colored?
Because they contain chlorophyll in them.
Pill. And what is its function? What's the function?
Leaves.
What's the function? Photosynthesis,
Leaves.
right? Where do you find them mostly? In the leaves.
The
Okay.
leaves. Yeah.
Yes. Yes. Yeah. This is the structure of one chloroplast. We don't study the structure of other one because it doesn't have any complicated structure. Just filled with protein, fat, or what's a uh, leucoplast that stores fat, elioplast, and leucoplast that stores starch. And what stores fat? Elioplast. Okay, so this is chloroplast. How many membranes here? Two. In mitochondria, how many membranes? Two. Rest all have single membrane. Where do you find no membrane? Ribosome. Okay, right. So there is outer membrane and inner membrane. Now, what's the difference between chloroplast and mitochondria? What's the difference? Yes, inner membrane is folded into what? Cristae. What's on the cristae? What do you find on the cristae? Stalk particle. And what's the function of stalk particle? Hmm. ATP formation. Okay, and even this has intermembrane space, but it doesn't have the same function of mitochondria. What's the function of intermembrane space? H plus. Okay, and what is jelly-like substance there? Matrix, and here it is called stroma. See, stroma. Matrix starts with M in mitochondria. Learn that way. Okay, this is in the chloroplast, stroma and chloroplast. Even this is a jelly-like substance. Okay, in photosynthesis, there are two reactions. Okay, study them in the other chapter. The first one is called light dependent reaction, or sometimes they call it light reaction. Okay, right? It's light dependent reaction or light reaction. In this reaction, the light energy is converted to ATP. Okay, how do we produce ATP? By respiration. How do chloroplasts produce ATP? From light. Okay. Second step. In the second step, it's called dark reaction in our syllabus. It's light, it doesn't need light. Dark reaction means light independent reaction. Okay. In this reaction, using ATP, using ATP, carbon dioxide become glucose. Okay, learn this one. What are the two steps of reaction? Light dependent. It's wrong to say dark reaction, but for the time being, it's okay, right? Light reaction and dark reaction. That's easy for now. When we do the other chapter, we'll change. What's a light reaction? It needs light. Dark reaction doesn't mean it happens in dark. It doesn't need light. Okay. What happens in light reaction? Light energy is converted to what? ATP. And dark reaction, carbon dioxide is changed to glucose using ATP. So, why light is required? Light is required to produce ATP. Make sense? Okay. And inside this, what do you find inside this? You can find many flattened structures known as thylakoid. Okay, disc-like structures, thylakoid. And inside the thylakoid, if you look inside the thylakoid, you find it contains chlorophyll. This is filled with chlorophyll. Okay, this is these are the pigments. One pigment will learn chlorophyll, but there are many more, which is in the other chapter. No, it's filled with chlorophyll, right? Okay. And so, what's the function of this? It traps energy from light. It takes energy from light and produces what? ATP. So what happens in the thylakoid? Light dependent reaction or light reaction. Where does light reaction happens? In the thylakoid. What is thylakoid? It's surrounded by membrane and it contains pigments like chlorophyll. What chlorophyll does? Absorbs light energy, change it into ATP. Okay, we have the whole reaction, how ATP is formed in a different chapter. So we'll study that later. And this is a stack of thylakoid. And what do you call a stack of thylakoid? This is called granum. What is granum? A stack of thylakoid. Why there are so many? To increase the efficiency. So more ATP, more light is taken in. Okay, and this granum is for one. Grana, grana means plural. Grana is more than one. Granum is just one of them. And you find hundreds of such grana. And they are interconnected by intergrana. Okay, intergrana. Okay, so they are connected by intergrana. Good? Right. And what do you find in the stroma? In the stroma, you find circular DNA. And you find presence of 70S ribosome. What does that indicate? Even this was a bacteria. Okay, which was, even today you find blue-green algae, cyanobacteria, something similar to that, entered plant cells. That's how plant cells evolved in the before 3 billion years. This happened. In the beginning, the this bacteria entered the eukaryotic cells, and then it developed the cell wall, and green color become plant cells, and they are known as algae today. Then algae moved on the land. Do you remember in the class 9 you studied bryophytes? Then it became pteridophytes and later gymnosperms and today's flowering plants. Okay, so that's a line of evolution. Anyway, is it similar to bacteria? Yes. Is it similar to mitochondria? Say similarity between chloroplast and mitochondria. One similarity. Two membranes. Say another similarity. Mitochondria and chloroplast. Circular DNA and the 70th ribosome. Any other similarity? Both have a jelly-like substance. What's a jelly-like substance here? Stroma. What's the other one called? Matrix. Good? Okay. And this is the structure of this is the structure of chloroplast. And what's the function of stroma? In the stroma, dark reaction happens. Okay. And where does light reaction happens? In the thylakoid. Okay. So let's review this. How many membranes around? Two. What do you call them? Outer membrane, inner membrane. What's the space between the two? Intermembrane space. Okay. There's no specific function, so we don't learn that. What's a jelly-like substance inside? Stroma. What do you find in stroma? Enzymes for what reaction? Enzyme for dark reaction. Okay, means what is dark reaction? Reaction which doesn't need light. It's called dark reaction. Okay, and what do you call this structure? What is this? Thylakoid. And name a group of thylakoid. Granum. Okay, and grana are interconnected by intergrana. Okay, intergrana. And function of grana. What's the function of grana? Light. Yeah, light reaction. Same. And what do you find in the stroma? Circular DNA and 70th ribosome. Plus enzymes for what? Dark reaction. Okay, what's dark reaction? Reaction which doesn't need light. It's called dark reaction. Okay, it has enzymes for dark reaction. Is it clear? So, how many membranes around this? Name them. And what's a jelly like substance inside? And what's the function of what do you find in stroma? Enzymes for dark reaction. What is dark reaction? Does not need light. Okay. And what's the function of thylakoid? Light dependent light reaction. What is inside the thylakoid? Chlorophyll. Chlorophyll. What does chlorophyll do? Yeah. Traps light and produce ATP. Okay. What happens in light reaction? Light changes to ATP. And in dark reaction, what happens in dark reaction? Need light. And what change happens in what's the reaction? Carbon dioxide become using energy from what? ATP. Is it similar to mitochondria? Yes. Why? Name two similarity. Membranes and hmm, 70th ribosome. Salome, what's the function of thylakoid? Function of thylakoid? Light reaction. Okay, light reaction. And what's the function of the granum? Salome.
Yes, sir.
Yeah. Where does
Half light.
light? Yeah. Where does light reaction happens? Light
uh,
reaction. Where does it happen?
in the granum.
In the granum, thylakoid. Where does dark reaction happens?
In the stroma.
In the stoma. Okay. Samira, what do you find in the stroma?
Um, jelly-like
Substance.
substance.
What reaction happens there? Dark reaction, right? What is dark reaction?
Um, in um, in the presence of ATP, CO2 is connected to glucose.
Glucose. Yes, very good. They are in your school, right? Um, Samira and Salome, you are in senior or Aman?
So now in senior, before in Amman.
Aman, okay. You're in Aman. Okay, that's a plastid, chromoplast. Okay, amylo, elio, proteinoplast. Okay, this is proplastid means it's an undeveloped plastid which turns into plastid. Etioplast is the second form. From this one, this is formed, and that turns to one of these. Okay, so that's explanation of this. Yeah, those are the in the mesophyll cells. What do you find? Chloroplast. Those are the chloroplasts. The sizes you have to learn, length and width. Okay, for the needs they always ask these sizes. Okay, that's a there you can see. What is this? Stack? No, granum, right? And what's a jelly-like substance inside? Stroma, right? Okay. And so lumen of thylakoid. Okay, all those things, right? That's ribosomes in eukaryotes. What's ribosome in eukaryote? ATS. What is it made up of? 60S and 40S. Okay, what's a large subunit? Large subunit is 60S and small subunit is 40S. Okay, and is there a membrane around? Is it covered by a membrane? No membrane. What is it made up of? RNA and protein. Okay, and what's the name of the RNA? Remember, rRNA, ribosomal RNA. Okay, and what is its function? What's the function of this? Protein synthesis. Where do you find this attached? In eukaryotes, where do you find this? On the endoplasmic reticulum. Okay, remember. So in eukaryotes, you find most of the ribosomes are on what? Endoplasmic reticulum. Okay, and what are the two subunits? Large subunit and small subunit. That's easy, right? What is ribosome made up of? Protein. Do they have a membrane? Yes or no? No. Okay. What kind of ribosome is in eukaryotes? Mhm. 8 years. And what are the two subunits? 6 and 40S. And what's the function of this? Yeah. And where do you find them attached all the time? Reticulum. What do you call that endoplasmic reticulum? With the ribosomes? Hmm. Rough. Rough endoplasmic reticulum. Okay. There you can see. See, what makes them different? These are the RNAs. Like if it's prokaryotic. It's 70s, right? 70s ribosome has 50s and 30s. And see, these are the RNAs. These are the RNAs and 34 proteins in the large one. In the small one, okay, one rRNA and 21 protein. But so see the difference here. When it's 60 years, you find these are the proteins and more number of proteins become heavier. And this is the protein. So what make them different? The number of rRNA and number of protein. That's it. Don't have to learn that. Okay, this is translation. What is translation? It's a protein synthesis. But exactly this step is called translation. But that's in class 12. And protein synthesis is a function of ribosome. And during protein synthesis, the RNA, mRNA comes in and the protein is formed. This is the protein. Okay, learn it as protein synthesis. Don't learn this now. That's a ribosome. Where do you find them sticking? On the endoplasmic reticulum. Okay, look at this guy. He is one who discovered this and to give you trouble to memorize all this thing, right? He's the first one to observe the ribosomes. Okay, this is called cytoskeleton. You know, in a cell, uh, this is an animal cell. Imagine all the parts of the cell are simply, floating inside. What will happen to the parts of the cell? Some of them heavier ones will go to one side, settle down, pile up over each other, isn't it? Okay, to prevent that, there is a skeletal system inside the cytoplasm. This is a cell membrane, that's a cell membrane. And inside the cytoplasm, can you see a skeleton? That this skeleton is called cytoskeleton. What is cytoskeleton? It's a skeletal system inside. Do we have skeletal system? What's our skeletal system made up of? Bones. Can you imagine yourself without bones? You'll be like heap of, yeah, we'll be moving like amoeba. We can't, can we stand up? No. Our muscles can't function, right? Similarly, every organelle is in one place. See, they're fixed in one place. They're not simply moving all the time, right? And this system is called the cytoskeleton. Cytoskeleton is made up of different kinds of filaments. Those are the filaments. Can you see? Intermediate filament, microtubule, and actin filament. These filaments form cytoskeleton. They help in supporting the cell, even help in movement of the cell. Okay. What is cytoskeleton? A network of filaments, protein filaments in the cytoplasm. And these protein filaments give mechanical support, help cell to move, and maintains the shape of the cell. Three things. All right. Is there another picture here? No. Okay, so what is cytoskeleton made up of? Okay. Cytoskeleton made up of protein filaments. Okay. Cytoskeleton is made up of protein filaments. Okay. Some of the protein filaments are microtubules. This is, do you remember microtubule? In the flagellum. Okay. Microtubule. And microfilaments. And intermediate filaments. The three kinds of filaments. What are they? Microtubule, microfilaments, intermediate filaments. Okay, these are three types of filament. And what's the function of cytoskeleton? It gives support, mechanical support. It gives shape to the cell and help the cell to change the shape. When do the cells change the shape? Do you remember this? Phagocytosis, engulfing bacteria. The cell has to change the shape. And that's done with the help of the cytoskeleton. So, what is cytoskeleton? Protein filament in the cytoplasm. Okay, what are they? Protein filaments in the cytoplasm. And it's made up of three types of protein filaments: microtubule, microfilaments, and intermediate filaments. Okay, microtubule, microfilaments, and intermediate filaments. Okay, then what is its function? It gives support to the cell. Okay. And it gives shape to the cell. Every animal cell has a different shape, like muscle cells, everything has a different shape. And it helps cells to change the shape. Is it clear? Okay. What is cytoskeleton? Cytoplasm. Why is it there? And what is it made up of? Yeah, microfilaments and intermediate filaments. And why is it there? Support, shape, and change the shape. And can you give an example when the cell changes the shape? Phagocytosis. Do you remember how amoeba moves? Pseudopodia and these things. Okay. So these are the three functions of the cell. I mean the cytoskeleton, right? Yeah. Salome. What is cytoskeleton? What is cytoskeleton?
They are the protein filaments
Yeah.
present in the cytoplasm.
Yes. And can you name them? The protein filaments?
Microtubular, microfilament,
Tubule, filament, and intermediate
intermediate
filament.
filament.
Okay. Samaira. What's the function of these?
This is mechanical support and electrical.
Yes. Okay. Right. Now we'll study about cilia and flagella. Want a break? 5 minutes break you take. It refreshes your mind. Take 5 minutes break. Stay there. Don't run away. Samaira and Salome. Stay there, okay? will start on Monday? No, Tuesday. Every class. You started. Why are you using glass? You use mobile too much from childhood. You read a lot. Then it's mobile, right? Play game. 0.2. Stay. Stay. But you don't have that. It's a screen you stay inside. How do you read? You sit like this, sit upright. Yeah. Sit close, right? Always sit upright. Buy a good chair. Till your time. The office chair. And sit upright. Buy a stand. Like to keep mobile stand. Book stand. Keep it like this and read. You can get it on. You know, there's exercises for the eye when you wake up. In yoga, they teach that to look up, down, sideways. Do you feel pain anywhere when you look sideways? Try both sides. No. And this is strain. It's a strain. You don't read any. Right? Only school book. I would. Well, school. Okay. But there are people in the class who read, right? They have glasses. So it can be screen, excessive of screen, or just both. But we are forced to use these classes are online. Again, it's increasing, right? You keep the screen away. Don't go close to protect the eye. And there's a glass they use for the PC, which cuts off blue light. Okay? Yeah. There's both power and filter, anti-glare. The celian flagella. Do you remember cilia? There is a picture of. No. Okay. Right. And where do you find cilia? You can see cilia in, yeah, no trachea. In the lining of trachea. I mean trachea, but lining of trachea, right? But this animal, the one you have seen just now, what's the name of this? Yeah, paramecium. The paramecium has short hair-like structures. This is cilia. Okay. How about the bacteria we studied? Bacteria has flagella. So what's the difference between cilia and flagella? Cilia you find many all the time, always. They are short, and flagellum are always long. You might find one, two, or few, very few. Okay, not too many. That this is always many. But inside the structure is the same. Both are made up of, can you see that? Microtubules. These are microtubules. But what's the difference here? 9 plus 2, 11. The 11 microtubules in both cilia and flagella. And both have a basal body, like in bacteria. So what's the difference between bacterial flagellum and flagellum of eukaryotes? Total microtubule 11. Other one 9, only 9. Okay, there's a difference, right? And there you can see. They call this axoneme, the core. This core is called axoneme, and this is the cell membrane, plasma membrane. There's a covering of plasma membrane, and inner part is known as the axoneme. It has like groups of 2, but 9 of them. See, 1, 2, 3, 4, 5, 6, 7, 8, 9, and 2 in the center. That's a difference between prokaryotes and eukaryotes. Eukaryotes have how many? 11 microtubule. Prokaryote has minus 9. Okay. And those details we don't need now. Okay, that's another structure. It's called centrosome, right? What is centrosome? Centrosome you find in animal cells. Animal cells contain centrosome. Okay, if you look at animal cells, it's an animal cell. You find the nucleus, and at one pole, you find one pair of centriole. How many? One pair. Okay, and it's surrounded by a clear zone of cytoplasm. And there, what do you find? Microtubules radiate from here. These are microtubules radiating from. Look at the arrangement. What's the arrangement? They are at right angles to each other. Always you find this arrangement. So how many centrioles? Two centrioles. Where do you find centrioles? Only in animal cell. They are absent or not visible in plant cells. So that's the difference between plant cells and animal cells. Can you say other differences? Yeah, plant cells have cell wall, animal cells don't have. Another one. Hmm. Chloroplast in plant cells. Okay, another difference. Large vacuole. Here, yeah. And this is the last difference. What's the next difference? Animal cells have centriole. When plant cells, it's absent. Okay. And what is radiating from here? What do you call this? Microtubule. Okay. The centriole plus the clear cytoplasm around it together form centrosome. So, what is centrosome made up of? Centrosome is made up of one pair of centriole and the cytoplasm around it is called centrosome. No membrane around it. So, name two organelle without membrane. Yeah. Ribosome and centrosome, right? What is centrosome? One pair of centriole with the cytoplasm around it. Okay. What's the function of this? Do you remember the mitosis? During mitosis, you will find two centrioles from opposite sides. They produce a fiber known as spindle fiber, and this attaches to chromosome. See, this connects to chromosome. This is the These are chromosomes, and they pull and split the chromosome. It was in class 9. Do you remember anything related to this? No. Okay. Anyway, so what's the function of the centriole? Centrioles form spindle fiber. What is spindle fiber made up of? Microtubule. And why they are formed? To separate chromosomes. This is to separate chromosomes. So we have a lesson on mitosis, a full lesson on mitosis. There we'll study in detail. Okay, right? So what's the name of this organelle? Centriole, right? Or centrosome. What is centrosome made up of? One pair of centriole, one pair. Okay, and how they are arranged at right angles to each other, right? Okay, and during mitosis or during cell division, during cell division, this duplicates and form one more, and it goes to the other side, right? That's why you find two there. And what do they form? Spindle fibers. When do they form spindle fibers? During cell division. Can you name the two types of cell division? Mitosis and meiosis. Okay. During mitosis and meiosis, they duplicate, move to the other side of the cell and forms spindle fibers. Spindle fibers are made up of microtubule. Microtubules separate chromosomes during cell division. Is it clear? Yes. So, what's the name of the organelle? And centrosome. Okay. What is centrosome made up of? Hmm. Cytoplasm. How come there are two here? Hmm. Duplicates into two. Okay. And what do they form? Spindle fibers. What is spindle fiber made up of? Microtubule. What's the function of microtubule? What do? They separate chromosomes. They separate chromosomes during cell division. Good. How many centrioles are there? One pair. Means two. Yeah. Right angles. In what kind of cells you find this? Animal cell? Animal cells. Okay. And what are spindle fibers made up of? Microtubule. Salome, what is a centrosome? What's a centrosome?
Centriole and cytoplasm.
Cytoplasm. And what do they form? What do they form? Spindle fibers, right?
Yes, sir.
Okay. Samera, what's the function of spindle fibers?
It's to separate the chromosome.
Chromosomes. Okay. Yeah. There you can see. That's one pair of centriole, right? One pair of centriole. And these are the microtubules on it. There's more detailed structure. Don't learn it now. We'll learn it later. Like that's what they're talking here. Right? Okay. That's a nucleus. Let's study about nucleus. In what kind of cells you find nucleus? What type? Eukaryote. Is there nucleus in prokaryote? What do you find in prokaryote? Circular DNA. What's the other name of circular DNA? Nucleoid. Okay, nucleoid. All right. So this is a nucleus. Even nucleus has two membranes. Nucleus is surrounded by two membranes. See, there are two membranes around the nucleus. Okay, what do you call them? Outer and inner. Outer membrane and inner membrane. And there's a space. What's the space called? Intermembrane space. This is called intermembrane space, right? Okay. And see, this is connected to endoplasmic reticulum. You will find rough endoplasmic reticulum attached to the outer membrane. Okay. And there are pores on the nucleus. What do you call this? Nucleus. Okay. Nuclear pores, right? Right? Nuclear. It's round in shape. My diagram is not good. Okay. It should be round. It's called nuclear pore. What's the name of the pore? Nuclear pore. Why there is nuclear pore? To allow, transport molecules in and out of the nucleus. And there is a cytoplasm inside. It's called nucleoplasm. What is nucleoplasm? The cytoplasm inside the nucleus. Okay. And this contains fiber-like structures. And they call this chromatin. What is chromatin made up of? DNA and proteins. What's the name of protein? Histone. DNA and protein together form chromatin. What's the function of chromatin? Chromatin forms chromosomes. It's a chromosome. So what's a chromosome? It's coiled up chromatin. See, when the chromatin coils up, it forms chromosome. When do they form chromosome? During cell division. What kind of cell division? Mitosis and meiosis. Okay, and there's a small nucleus inside. This is nucleolus. Function of nucleus, this is where ribosomes are formed. So where are ribosomes formed? In the nucleolus. Is it clear? So this is a structure of nucleus. How many membranes around? Two. Outer and inner. Outer is connected to rough endoplasmic reticulum. Okay. And what are the pores on it? What do you call the pores? Nuclear pores. And why is it there? To transport molecules in and out of the cell. And what's a cytoplasm inside? Known as nucleoplasm. Okay. And what do you call the fiber inside? Chromatin. What is chromatin made up of? DNA and histone. What is histone? It's a protein. Okay. And this chromatin forms chromosome. When do they form chromosome? During, yeah, by coiling up. When do they form it? During cell division. Okay. Structure of chromosome we'll study later, right? And this is the structure of nucleus. Can you explain nucleus structure? Hmm. Outer inner membrane. Yeah, attached, connected to. Yeah. And yeah. How about nuclear pores? Molecules in and out. Okay. And what's a fiber inside? Yeah. Say. Name the fibers inside. Chromatin. What is chromatin made up of? Histones. And what's the function of chromatin? Forms chromosomes, right? And what is this? What's the function of nucleus? Mhm. Ribosomes are formed. So where are ribosomes formed? In the nucleolus. Okay. And what is chromatin made up of? Histones, right? Samaira, what's the function of nucleolus? Function of nucleolus? Yeah. Where are they? Okay. Okay. So there you can see. This is nucleus. Okay. That's another name. Nuclear membrane is called envelope. Okay. Envelope is also used. And this is chromatin. And that's a nucleolus. Okay. Perinuclear space means intermembrane space. Okay. Periphery, outside. What is this? Rough endoplasmic reticulum. What are those, flower-like things? Nuclear pores. Why is it there? They are like proteins which control movement. Like, it's not like open. There are pores, pore protein. Okay, that's a chromosome. Okay, in a chromosome, you find a disc in the center. Okay, this disc is known as centromere. What's the name of the disc? Centromere. This part. This constriction is called centromere. Outside that, you find a protein covering. It's called kinetochore. On the kinetochore, the spindle fibers attach. See, these details maybe we'll do in mitosis. Skip for time being. Okay. So this is chromosome. Don't learn this part. We'll do it in mitosis. The same thing is repeat. Mhm. Yeah, this is centromere. The other one was centriole. Okay, and that's chromosome. Just learn it's a chromosome. And this types of chromosome. Okay, it all will do these things in the other chapter. Okay, these are microbodies. Microbodies are. Do you remember microbodies in bacteria too? What was it made up of? Maybe gas vacuole. Okay, and what else? Sometimes glycogen. Okay, similarly, these are like extra things which is stored in the cell. This is called microbodies. Okay, it has one membrane around it and a crystalline core. Mostly you find stored substances. Okay, minute vesicles that contains various enzymes present in both plant cells and animal cells. We don't. That's it. It doesn't have any detail. These are known as microbodies. So we have completed the chapter. Okay. Yeah. And it's a big chapter. You didn't realize it. Yeah. And find out what they are starting in your school. Next lesson, I'll teach you that. For you, they'll take 2 weeks to reach the finish this. So relax. Uh, then we'll go to whatever chapter you start in your school, right? I think it's from chapter one. Do they give you syllabus? Hmm. Yeah, your plan, right? Hmm. So you have a book or something? Diary? They'll tell you. Hmm. So you'll come, you come to know all the chapters for this term. Mm. Send me a picture. So we'll follow that, right? Yes. Okay. So those are difference between plant cells and animal cells. What are the differences? Cell wall present, absent. Plastids means maybe chloroplast, right? Everything. It's absent there. Large central vacuole, centrioles, right? Mhm. Yeah, animal cells have no plastid. That's fine. Don't say chloroplast. You say plastid. It can be any of this, Like class 9, we say chloroplast, but now you say plastid, right? Difference between eukaryotic cell. Size, small, big. And here it is nucleoid, circular DNA. There's nucleus and chromosomes, right? Nuclear membrane is absent, it's present. And organelle is absent, present. Circular DNA. And they're linear. What's a linear means? It's like one thread. Here, it's like it's ends fused together. Okay, linear DNA and ribosomes 70 years. There 8 years. Okay, but in eukaryote, do you find 70 years? Yes. Where? In the mitochondria. Remember that. But that doesn't mean animal cell is prokaryote. Why there is 70 years in mitochondria? Because it was originated from a bacteria. Okay, and it also has circular DNA, which is the bacterial DNA. Okay, right. Finished, right? Yes. Now, do the questions. Yeah, the one I sent you. And if there is anything which doesn't make sense, skip it. We'll discuss it here. Maybe next lesson we'll do questions, and then we'll start the new chapter. And we'll have a small test next week. Okay. All right. See you next Thursday. This timetable is okay, right? Friday, is it okay? 11 or 12? You have dance class. 2:30. Yeah. 12 is okay. Fine, right? When is your church? You don't have church. You have church, right? Yeah, but it's fine. Okay. You have no issues. Yes. Morning you're free. Okay. So is this recordings helpful? Yeah. Even otherwise, if you are lazy of memorizing, just go through it. It's easy. All right. Okay. See you next week. Have a good day, good week, and all the best for new session tomorrow. Yes. Okay. Bye, Salome.
Bye,
Samira.
sir. Thank
Bye
you.
bye. Bye.